¿Aló? ¿Sí? ¿Se escucha? Comité que me diga, por favor, si se escucha.
Todo en
Sí,
orden.
Muchas gracias. Daremos unos pocos minutos para seguir aceptando más invitados y luego comenzaremos con la bienvenida. Muchas gracias por esperar. ¿Aló? Sí. Buenos días a todos los presentes. Mi nombre es Pablo Jiménez y los acompañaré como su anfitrión durante el transcurso de esta actividad. Preside esta ceremonia el señor rector Dr. Juan Manuel Solesi, CID. Asiste en su representación el vicerrector de apoyo al estudiante Dr. Bernardo Morales. Saludamos a las autoridades de la Facultad de Química y Biología que nos acompañan esta mañana. La decana, doctora Leonora Mendoza, la vicedecana de Docencia y Extensión, doctora Brenda Modak, la directora del Departamento de Biología, doctora Milena Cotoraz, y la jefa de carrera de Bioquímica, doctora Marcela Víquez. Cuerpo académico de la Facultad de Química y Biología, directiva NED, miembros de la comisión organizadora, compañeros y compañeras de bioquímica y otras carreras afines. Amigos y amigas y público espectador presente, es un agrado poder saludarlos. En nombre de la comisión organizadora es que les damos la bienvenida a este primer congreso virtual de estudiantes de bioquímica, organizado por los estudiantes y socios de bioquímica de la Universidad de Santiago de Chile. El tenerlos todos aquí nace desde una idea de hace tan solo poco más de un mes. Una idea muy prometedora, pero bastante poco pulida. Generar un espacio de divulgación científica y recaudar fondos para las familias que lo están pasando mal debido a la pandemia. Un proyecto por gestionar entre un pequeño grupo de buenos amigos, basado en los pilares de nuestro rol como científicos, y uno de los aspectos más compartidos en estos últimos tiempos, la empatía de ayudar a quienes más lo necesitan sin esperar nada a cambio. Solo bastó una idea consolidada por los correctos principios para hacer crecer este proyecto. Y el día de hoy nos encontramos aquí presentes gracias al arduo trabajo de una comisión organizadora, un gran equipo que se vio apoyado por diferentes personas que sin ellas esta actividad no sería lo que es ahora. Es por ello que queremos agradecer a todas las personas y entidades cuya colaboración hizo posible la realización de este congreso. Agradecemos a los miembros de la comisión organizadora, los socios ANEF Filial USACH, por la iniciativa de generar esta instancia de divulgación científica sumamente necesaria dentro de este contexto. Al apoyo y difusión brindado por las autoridades de la Facultad de Química y Biología, doctora Leonora Mendoza, doctora Brenda Moda y doctora Marcela Wilkens, quienes vieron la importancia de la realización de una actividad así y se comprometieron en todo lo posible por el buen desarrollo de esta. Al equipo de funcionarios de la Facultad de Química y Biología, Nicolás Gaona, periodista y encargado de comunicaciones, Roberto Castro, encargado de laboratorios de computación, Jaime Quiroga, jefe de área informática, y Patricia Jara, secretaria de decanato, quienes brindaron su ayuda en muchas de las tareas implicadas en adaptar un congreso a este formato virtual en el que nos encontramos. Al grupo de divulgación científica Nuevo Foco, por toda la ayuda brindada en la difusión de este congreso, la generación de un foro dentro de las actividades y la cobertura periodística durante el transcurso de esta semana. A las agrupaciones de ayuda humanitaria, El Bosque Ayuda, Peñalolén Solidaria y La Olla de Puente, por su vinculación con esta actividad, cuyo pie inicial es y ha seguido siendo brindar ayuda a las familias que más lo necesitan en estos duros momentos. Y finalmente agradecemos a todos los asistentes de este congreso, cuyo interés e incremento día a día fue uno de los mayores pilares de motivación para realizar y poder brindarles hoy un congreso de calidad. Estamos muy agradecidos a todos ellos. Invitamos a nuestra decana, la doctora Leonora Mendoza, a compartir unas palabras de bienvenida.
Muy buenos días. Estimadas autoridades de la universidad, representadas por el vicerrector de apoyo a los estudiantes, doctor Bernardo Morales, y de nuestra facultad, representadas por la directora del departamento, la vicedecana de docencia y extensión y la jefe de carrera de la carrera de bioquímica, que hoy día nos acompañan. Un saludo especial a los académicos y académicas de otras instituciones y de la Facultad de Química y Biología, y en particular a todo el estudiantado que está presente. Mis primeras palabras al inicio de este primer congreso virtual de estudiantes de bioquímica y carreras afines es para las y los estudiantes participantes en este congreso y muy especialmente para el equipo organizador del evento, para el que con mucho interés y orgullo hemos colaborado y acompañado como Facultad de Química y Biología. Estoy segura que este congreso virtual será un éxito y que es además una muestra más de la tremenda capacidad de nuestro estudiantado para relevar la ciencia aún en el contexto en que nos encontramos. Primero que todo, les deseo que disfruten y aprovechen las actividades que están programadas durante esta semana, que sé que las y los estudiantes han organizado con mucho ahínco y cariño. En segundo lugar, quiero felicitarlos por esta iniciativa que ha significado no solo discutir temáticas científicas más relevantes de la actualidad, sino que también por preocuparse por las personas y las familias más afectadas por esta pandemia. Es por esto que esta noble iniciativa refleja el sello de la Universidad de Santiago de Chile y sus valores, que tienen que ver con la solidaridad y la permanente preocupación por aspirar a una sociedad más justa. Siendo formada en esta universidad, personalmente estoy convencida de que este tipo de iniciativas se deben aprovechar al máximo. Pese a las dificultades que se presentan, este congreso es una oportunidad de crecimiento y de formación profesional que, dada la contingencia, contribuye a complementar conocimientos, generar discusiones y profundizar en temas de interés general. Por último, destaco el hecho de posibilitar el acceso a charlas, mesas y paneles de manera gratuita como una forma de acceder a la ciencia a las personas que hoy, debido a la pandemia, tienen un mayor interés por conocer sus alcances y sus soluciones. Agradezco desde ya a los charlistas extranjeros y nacionales que aceptaron este desafío. Finalmente, felicito nuevamente a toda la organización que ha generado este espacio y también a todas y todos quienes están participando y han hecho posible esta actividad, en especial a todo el equipo de profesionales de nuestra facultad que con mucho compromiso ha apoyado la realización de este congreso. Disfruten de esta jornada y muchas gracias por ser parte del congreso. Que tengan una excelente semana. Muchas gracias.
Agradecemos las palabras de la doctora Leonora Mendoza. Hace 2 años, por estas mismas fechas, nos encontrábamos ante uno de nuestros mayores desafíos y logros a obtener, la gestión del 35º Congreso Nacional de Estudiantes de Bioquímica, una instancia en la que con un año de arduo trabajo logramos tener a importantes exponentes ante un público de más de 400 socios inscritos. Ahora, en un nuevo y aún más adverso contexto, nos sentimos sumamente orgullosos del trabajo que hemos logrado. Miembros de ese antiguo comité se han mantenido y otros nuevos se han ido sumando. El fruto obtenido por este nuevo equipo de trabajo ha sido esta instancia de divulgación científica y apoyo humanitario, cuya idea nació hace menos de 2 meses, y hoy, teniendo importantes exponentes en el auge actual y con 750 inscritos interesados, estamos sumamente satisfechos del trabajo realizado. Queremos invitar, en representación de este equipo de trabajo, a la señorita Patricia Romo, miembro del comité organizador y delegada ANEP de la Universidad de Santiago de Chile. Le damos la bienvenida.
Buenos días a todos. Quisiera comenzar agradeciendo la asistencia de las autoridades presentes al primer Congreso Virtual de Estudiantes de Bioquímica y Carreras Afines, especialmente al vicerrector del apoyo al estudiante, el doctor Bernardo Morales, a nuestra decana, la doctora Leonora Mendoza, nuestra vicedecana, la doctora Brenda Mudas, y a nuestra jefa de carrera, la doctora Marcela Wilkens, quienes desde el primer día en que comenzó la organización del Congreso nos expresaron su infinito apoyo. Agradecer la asistencia a los estudiantes de bioquímica de distintas universidades del país e incluso a algunos que se encuentran fuera del país, a los estudiantes de carreras afines y no tan afines, y además a los futuros estudiantes de carreras científicas, porque no has confiado la oportunidad de abrir los límites de tu conocimiento. Y espero que todos salgamos de esta semana sabiendo un poco más. Pero no solo eso, espero que todo este congreso los motive a seguir informándose de distintas áreas, porque no hay nada más bello que seguir aprendiendo. Hoy nos encontramos en una situación excepcional. La pandemia nos ha afectado a cada uno de nosotros de manera física y emocional. Tengo más que claro lo difícil que es sobrellevar todo lo que estamos viviendo. Y deseo de todo corazón que cada uno de ustedes pueda salir adelante junto a sus seres queridos. Y el día de mañana podamos reencontrarnos en la universidad, en el parque o en la calle, y podamos romper esta distancia social que nos ha mantenido a todos alejados, de manera física, pero no de manera emocional, porque hemos encontrado la manera de seguir juntándonos y poder darnos apoyo a pesar de las distancias. La gente de todas partes del mundo ha empezado a darle la importancia que se merece la ciencia. Tuvo que suceder una crisis sanitaria para que esto ocurriera, y esperemos que una vez terminada esta pandemia, la importancia que difícilmente consigue obtener la ciencia no sea olvidada y desechada hasta una próxima crisis, de que junto con la necesidad también sale a la luz las distintas falencias que conlleva la falta de desarrollo científico. Y no solo eso, la necesidad de que la población sea educada con información básica, como por ejemplo, ¿qué es un virus? Quien no se dedica a la ciencia también necesita entenderlo, y no solo necesita entender ciencia, necesita arte, historia, filosofía y política. Espero que se den cuenta desde el Ministerio de Educación que en las aulas no solo se necesita lenguaje y matemáticas, se necesita formar a personas con cultura y pensamiento crítico. Lo único que nos ha llevado la falta de educación en diversas áreas es aumentar la desinformación y que se vea tanta información falsa circulando por las redes. Es ahí donde también entra nuestro rol como futuros científicos o quienes ya son científicos. No solo puedo criticar a la educación de hoy, porque también tenemos una responsabilidad social. Debemos ayudar a romper la desinformación, a divulgar lo que sabemos y a ayudar a quien no comprende. No podemos encerrar la información en papers llenos de tecnicismo que una persona que no está vinculada con el vocabulario científico no entienda. No podemos encerrarnos en la academia y en los laboratorios, siendo que tenemos mucha información que otro puede necesitar. Es por ello que nosotros decidimos que este congreso sea público, porque queremos ser parte de la solución. Queremos romper con las barreras de la información y que todos puedan aprender temas que son sumamente importantes para la situación en la que nos encontramos, como lo son los avances del COVID-19, el cambio climático y nuestra propia salud mental. Hace más o menos un mes, un grupo de estudiantes de bioquímica se dio cuenta que la pandemia nos había quitado una gran herramienta en nuestra formación académica. Era el Congreso ANEP. En él conocíamos distintas áreas científicas en las que nos podíamos desarrollar. Pero es increíble cómo un lugar de confianza le quita los límites a tu idea. Se pensó en un ciclo de charlas para que sus compañeros conozcan nuevas áreas en donde desarrollarse. Un acto pequeño para suplir la falencia. Y así se generó una red de estudiantes que querían apoyar la causa. Pero conforme se recibía más apoyo, la idea crecía aún más. De 3 días pasamos a una semana. De un par de charlas pasamos a simposios, foros y talleres. E incluso nos cuestionamos por qué limitarnos solamente a nuestros compañeros si sabemos que todos necesitamos seguir aprendiendo. Y para ese momento yo ya estaba asustada. No sabía cómo habíamos llegado tan lejos y todo lo que nos proponíamos teníamos que hacerlo en contrarreloj. Pero hoy agradezco a cada uno de ustedes que dieron todo de sí para que esto funcionara. Porque teníamos metas muy altas, pero aún así lograron superarlas. Y aquí nos encontramos difundiendo ciencia como futuros científicos, cumpliendo nuestro rol. Pero saben, debo felicitarlos porque lo hicieron sumamente bien, porque lo difundieron a más de 750 personas. Y espero de todo corazón que el futuro que les viene por delante sea próspero, porque tengo claro que todas las metas que se propongan las podrán lograr. También quiero agradecer a don Roberto Castro y a Nicolás Gaona, quienes estuvieron codo a codo con nosotros proponiendo ideas y dándonos apoyo en las distintas necesidades que a nosotros nos surgían. Y para finalizar, quiero comentarles que uno de los objetivos de nuestro congreso no es solamente difundir ciencia, también es ayudar. Quiero comentarles de la campaña Beneficencia, porque sabemos que todos hemos sido afectados por esta pandemia, pero hay familias que necesitan de nuestro apoyo. Y es por eso que quiero invitarles que si quieren y puedan dar una donación a esta campaña para que en esta ocasión podamos ayudar a familias de Peñalolén, Puente Alto y El Bosque. Muchas gracias y bienvenidos al primer Congreso Virtual de Estudiantes de Bioquímica y Carreras Afines.
Agradecemos a nuestra delegada por su discurso. Antes de dar pie a nuestra charla inaugural, queremos dar un espacio de sumo respeto para homenajear a alguien que ya no se encuentra con nosotros. Con fecha de 17 de julio nos deja don Dr. Claudio Vázquez, académico de nuestra facultad desde 1983 y uno de los mejores docentes que miembros de este comité tuvieron el privilegio de conocer. Un verdadero ejemplo de compromiso con lo que respecta a la formación de estudiantes. Un profesor lleno de sabiduría y amabilidad, dispuesto a compartir lo mejor de sí a la hora de comenzar una clase. Queremos invitar al profesor Dr. Felipe Arenas a compartir unas palabras de conmemoración y agradecimiento por todos sus servicios brindados, a nombre de los estudiantes de esta facultad, sus colegas de trabajo y amigos personales. Miembros de este comité se hacen presentes como encienden sus cámaras como acto de presencia.
Hola, bueno, buenos días a todos. Primero que todo, quiero saludar a las autoridades presentes, tanto de la universidad como de la facultad, y sobre todo a los estudiantes organizadores de este congreso. Creo que una importancia muy, una organización muy importante para difundir la ciencia, y creo que la invitación que ustedes me hicieron para poder homenajear a don Claudio, que fue un académico destacado en la universidad y sobre todo fue un muy buen profesor, ¿ya? Yo preparé unas pequeñas diapositivas que me pidieron eh, en relación a Claudio para mostrar y comentar más que nada algunas cosas en relación a cómo era él como persona. Y como muchos estudiantes y como mucha gente supo y conoció a Claudio, él más que nada fue un científico, ¿ya? Él dedicó su vida a la ciencia, partió como todos que ustedes están partiendo, como muchos hemos iniciado y hemos logrado, estudiando bioquímica, una carrera que él consideraba que era muy bonita. Él se dedicó siempre a la investigación. Claudio fue una persona que desde sus inicios eh, partió estudiando bioquímica. Él estudió bioquímica en la Chile, se recibió en el año 77, y él siempre dijo, y las conversaciones que tuvimos, porque la gracia que tuve yo por lo menos fue que conocí a Claudio hace más de 16 años. Yo llegué a trabajar al laboratorio de Claudio en el año 2006, luego a terminar mi tesis de pregrado de bioquímica en LUSACH, con el profesor Emilio Cardemil. Y justo llegué a trabajar y conocí a Claudio ahí, fui a trabajar como ayudante de investigación, y posteriormente me quedé ahí trabajando. Ya después hice un doctorado, después un postdoctorado, y posteriormente pasé a ser colega de Claudio. Gracias a él pude ser contratado a la universidad, por lo tanto puedo hablar con mucha conocimiento de Claudio, que él siempre le gustó la bioquímica. Él, su pasión siempre fue investigar, conocer, pero siempre tuvo un carisma que fue muy importante, que fue que él quería hacer de la ciencia algo divertido, algo simpático, algo que él le apasionara, y que más que nada él quería tratar de descubrir cosas nuevas, ¿ya? Posteriormente Claudio estudió un doctorado en ciencias biológicas en la Universidad Católica, se tituló el año 83, ¿ya? Y Claudio inmediatamente quiso seguir en la academia. Claudio, para los que no saben, y mucha gente o bioquímicos que están presentes acá, y colegas y amigos, Claudio se fue a trabajar, primero partió trabajando en LUSACH en los años 83, trabajó 2 años en LUSACH, posteriormente trabajó 3 años en la Universidad de Chile, estuvo hasta el año 88 en la Chile, y posteriormente emigró él a trabajar a la Universidad de Talca. Y en la Universidad de Talca él estuvo alrededor de 7 años, hasta el 95, y después retomó a la USACH, volvió a la USACH a trabajar hasta este año. Y Claudio siempre nos contaba que él dijo que se puede hacer ciencia en Chile y investigar y saber conocimiento teniendo los mínimos recursos. Claudio siempre nos contaba, una de sus anécdotas era que él cuando se empezó a mover de sus laboratorios a trabajar de la USACH a la Chile a Talca, él se movía con una gradilla, con tubitos que eran sus bacterias y sus enzimas de restricción, y eso era lo único que él tenía, ¿ya? Y con eso Claudio de a poco fue forjando su línea de investigación, y Claudio decía, nunca fue fácil, siempre vamos a tener distintas trabas y distintos problemas para tratar de solucionar. Pero Claudio, en todo su momento que fue trabajando como científico, él fue sin querer queriendo diciendo, siempre él decía, yo no tengo idea de lo que estoy haciendo, de a poco voy descubriendo mi camino. Y Claudio logró ser uno de los investigadores muy destacados en Chile, en bioquímica y sobre todo en el área de microbiología que él se especializó. Claudio nunca fue una persona preocupada de cuántos papers tengo, de cuál es mi productividad científica. Él lo que hacía era investigar. Y para él bonos eran tener publicaciones científicas, pero lo que a él más le importaba era formar profesionales. Claudio logró formar una gran cantidad de profesionales, más de un centenar de profesionales, entre pregrado y posgrado, principalmente bioquímicos de la Universidad de Santiago de Chile. Así que Claudio fue capaz de formar generaciones. Claudio era un profesor que trataba a todos por igual, fueran autoridades, fueran alumnos, funcionarios. Claudio era una persona muy humilde y es algo que transmitió siempre a toda su gente. Todas las personas, yo puedo dar por firmado, que han pasado por su laboratorio fueron capaces de tener un legado de Claudio, que fue esa alegría y esa sencillez y esas ganas de investigar. Claudio siempre nos decía que él quería, él nunca se preocupó de los papers. Él, la única meta que una vez me contó, que yo supe que él estuvo preocupado de los papers, que cuando partió en esto, tenía uno de los profesores de él, que siempre le dijo, tu meta tiene que ser que cuando te jubiles tengas por lo menos el mismo número de publicaciones. Y Claudio siempre ese fue su objetivo, me dijo a mí. Pero si uno se da cuenta, y Claudio incluso él nunca contabilizó todos sus papers, y nosotros ahora viéndolo, Claudio logró tener cerca de doblar su edad en publicaciones. Claudio hasta el momento tiene más de 120 publicaciones en revistas científicas. Y Claudio lo hacía por amor a la ciencia, ¿ya? Claudio formó un legado tan grande que nosotros nos autodenominamos los telurianos. Él nos decía los telurianos, porque Claudio comenzó su línea de investigación y se desarrolló finalmente en el laboratorio de microbiología molecular, el que está acá. Acá está Claudio. Esto debe haber sido hace como 3 años atrás. Claudio siempre estuvo enseñándonos a todos, estudiantes de pregrado, posgrado, unidades de investigación. Claudio era el que transmitía el conocimiento a los alumnos, siempre tenía sus datitos para poder purificar de mejor manera el DNA, una proteína, siempre estaba con la bioquímica clásica tratando de entregar todo ese conocimiento. Pero sobre todo Claudio empezó a trabajar en un problema que él no conocía, que era el telurito. Si ustedes, mucha gente no lo conocen, no conocen lo que es este elemento, muchas veces, de hecho nosotros íbamos a congresos y éramos las únicas personas que trabajábamos con este elemento. Claudio llegó a trabajar con telurito de una manera más o menos anecdótica. Claudio partió trabajando con enzimas de restricción, harta biología molecular, después se fue a trabajar con termófilos, y un día encontró un organismo termófilo y empezó a plaquearlos en medios selectivos y encontró que se formaban coloración negra. Entonces él dijo, como le gustaba la enzima, dijo, voy a tratar de investigar qué es lo que hace, cuál es la proteína involucrada en este fenómeno. Y empezó a trabajar en esto y se dio cuenta que no era un problema tan sencillo, que la toxicidad o la resistencia que presentara algún organismo a este metaboloide era medio complicado. Entonces él dijo, como investigador, dijo, bueno, voy a tratar de cambiarme un poco de tema. Se fue a trabajar con el Helicobacter pylori, postuló uno de los proyectos, él dijo, no me lo gané, sí que dijo, voy a volver a lo que yo me manejo, y desde ahí, ya hace más de 25 años que Claudio lleva trabajando con el telurito de potasio. Y Claudio fue uno de los investigadores capaces de ganarse proyectos Fondesit continuamente, y un elemento tan sencillo a él le abrió una línea de investigación tan amplia. De hecho, Claudio podemos decir que fue o es el único investigador en el mundo que se dedicó 100% a este compuesto. Porque si nosotros vemos en un tema que no es muy candente muchas veces, y él siempre nos decía, el telurito no es un tema que está en boca de todos. Sin embargo, en el mundo existen 3 o 4 grupos de investigación que trabajan con este compuesto, pero asociado a otros metales. La gracia de Claudio, que Claudio era el único que trabajaba solo con el telurito de potasio, el que le dio todos estos frutos. Claudio tuvo, yo creo, la felicidad en su carrera de lograr poder descubrir una gran cantidad de datos que le permitieron dilucidar muchos mecanismos. Y yo creo que eso es lo que nos tenía más contento a Claudio, que cada vez que nosotros postulamos un proyecto, ganábamos algo, él se sentía feliz de poder contribuir. Y su contribución siempre era hacia todos los alumnos, la gente que estaba presente. Claudio no era una persona egoísta que él se le ocurría a él la idea y él trataba de formular un proyecto. Claudio siempre nos decía, el laboratorio somos todos, ¿ya? Y eso siempre quiso transmitirlo porque todos siempre tratamos de ayudarlo en el proyecto. Claudio era una persona muy amigable, siempre lo pasábamos muy bien con él, siempre nos invitaba y siempre que había que postular a proyectos nos decía, ya, aquí nos juntamos todos, íbamos incluso estudiantes que venían llegando al laboratorio, estudiantes de doctorado, postdoctorado, y hacíamos estas tormentas cerebrales que él decía, Claudio, que le gustaba juntarse a conversar con todos, porque él decía, hasta una persona que quizás no tiene todos los conocimientos puede aportar desde otro punto de vista y puede tener esa chispita de la idea que necesita él investigar. Por lo tanto, hicimos mucha investigación con Claudio y la gente que ha trabajado con él ha estado muy contento porque Claudio nos permitió formar parte, no solo metiendo las manos ahí con lo que él nos decía, sino que nosotros dar nuestra idea. ¿Qué otra cosa importante, Claudio? Es que Claudio, todos los que tuvieron clases con él o que lo conocieron, era una persona muy simpática, ¿cierto? Todos siempre tenemos muy buena, que llegaba a clase a la vieja escuela con plumón y pizarra. Él hacía todas las vías metabólicas, se las sabía de memoria, todas las estructuras. Entonces él siempre impartió su clase tratando con esa chispita de ser gracioso y hacer que la ciencia fuera entretenida, no más que leer un libro o ver una diapositiva, sino que Claudio mostraba esa imprima en el cual te hacía querer entender más gracias a pasarlo bien. Claudio siempre estaba en el laboratorio, como lo vemos aquí en la foto de la derecha, tratando de ver qué estábamos haciendo, tratar de molestarnos un poco, nunca estaba tranquilo en su oficina, siempre estaba con nosotros ahí. ¿Qué otra cosa, Claudio? Era tan simpático, tan buena onda, que siempre le costaba, yo creo que lo que él más sufrió desde el punto de vista académico era cuando tenía que retarnos. Porque Claudio era un profesor que no le gustaba retar a la gente. Él siempre trataba de dilatar lo que más pudiera los asuntos porque no le gustaba pelear con la gente o tener este tema de autoridad sobre los alumnos. Pero él, aquí vemos una foto que le saqué sin que se diera cuenta cuando nos estaba retando a todos en una reunión de laboratorio, pero su reto era 1 minuto y después no podía dejar de terminar el reto sin echar una talla, sin echar a reír, para que nosotros estuviéramos más tranquilos. Por lo tanto, Claudio siempre fue una persona que imprimió mucho cariño y amistad, ¿ya? Y no solo en el laboratorio, sino que también fuera de él. Yo creo que toda la gente, todos los telurianos, todas las personas que lo conocimos, compartimos con Claudio más que nada fuera del laboratorio. Y el Claudio nos enseñó que independiente de tus características, tu grado académico, Claudio, para todos éramos personas y compartía con todos. Era muy querido de los animales, siempre estábamos ahí en los asados dándole un pedacito de carne a los animales que andaban por ahí. Claudio siempre fue la persona que nos formó y nos ayudó a crecer como persona. Por eso todo el cariño que todos le tuvimos a Claudio. Claudio siempre nos decía, la ciencia no se hace solo en los laboratorios, también tenemos que hacer cosas extraprogramáticas, que es lo que más le gustaba a él. Claudio, que decía que el laboratorio era su segundo hogar, Claudio siempre nos invitaba a hacer nuestras reuniones de laboratorio para ver cómo íbamos, hacíamos reuniones, nos íbamos a la playa, le gustaba salir. El viaje más largo nos fuimos una semana a un retiro a Puerto Vara con todo el laboratorio, estuvimos ahí tratando de ver cómo avanzar y apoyar a todos los alumnos. Por lo tanto, Claudio fue una persona que logró generar que todas las personas que él formaran tuvieran una conexión que no se borrara en el tiempo. De hecho, desde que yo entré al 2006 al laboratorio hasta el día de hoy, todas las personas que han pasado por el laboratorio estamos en contacto, porque Claudio fue una persona que era capaz de cautivar a la gente, a tal punto que Claudio, yo una vez le pregunté, Claudio, ¿tú alguna vez has puesto algún letrero que solicitando tesista o alguien que venga al laboratorio? Porque me llamaba la atención que siempre había gente. Tiempo, con años buenos, teníamos más de 20 personas trabajando en el laboratorio con un solo tema. Y Claudio me decía, no, nunca yo he puesto un letrero. Y era porque siempre se iba el boca en boca, Claudio era tan cariñoso y que siempre llegaba gente a trabajar al laboratorio. Claudio era un ambiente tan grato que siempre que cautivaba a los alumnos que llegaban a la unidad de investigación, no salían más, incluso hasta después del doctorado. Por lo tanto, Claudio tuvo ese carisma. Y Claudio fue muy bien reconocido, él lo sabía. Él no buscaba que fuera reconocido, pero él sabía que logró muchas cosas en su carrera. Claudio salió no sé cuántas veces mejor académico del departamento, premiado en la facultad o en la universidad. Gracias a Dios logramos hacerse un reconocimiento a Claudio en vida, aunque él no quería, él no es del que le gustaba decir, reconocer lo que había hecho, pero Claudio fue muy querido y se notó sobre todo, ahora que falleció, las condolencias me llegaron a mí y puedo decirles que yo con mucho cariño recibí cerca de 70 correos de personas, alumnos, autoridades, funcionarios, amigos, diciendo lo cariñoso que fue Claudio y cuánto lo van a extrañar. Y con eso Lo único que puedo decirle y que Claudio siempre quiso transmitir a todos los estudiantes de bioquímica, traten de ser personas, hacer ciencia de la forma más sencilla posible, que su pega les guste. Él decía, bioquímica es una de las carreras más hermosas porque tú puedes pensar y solucionar casi todos los problemas que se te pongan en la cabeza. Por lo tanto, siempre Claudio decía, no dejen de pensar, siempre traten de una manera sencilla, humilde, de buscar la solución a un problema. Por lo tanto, nosotros con esta última diapo quiero agradecer a Claudio y aquí mostrarle parte del legado de Claudio que hizo durante sus 37 años, en el cual formó una gran cantidad de profesionales, de los cuales son muchos doctores y de los cuales muchos están formando a otros bioquímicos o profesionales de carreras afines. Por lo tanto, nosotros siempre seguimos con el legado de Claudio y espero que en el laboratorio nosotros sigamos por mucho tiempo formando bioquímicos de la manera que él pudo formar, ¿ya? Con su gran carisma, ¿ya? Y Claudio siempre nos decía, muchachos, una de las frases típicas que puedo nombrar aquí es, muchachos, ¿qué nos deparará el futuro? ¿Ya? Siempre pensando qué podía hacer él en la investigación. Claudio fue una gran persona, realmente estamos todos muy dolidos con la partida de él, pero a la vez estamos muy contentos porque nos dejó un legado muy lindo y que espero que todos los bioquímicos que han pasado y futuros que nosotros formemos en nuestra facultad puedan tener parte de Claudio y eso. Bueno, agradecer a todos y yo sé que han pasado un par de semanas, estamos todos muy sentidos. Y finalizar que yo creo que Claudio estaría muy contento si con este congreso que están organizando. Él siempre quería mucho a los bioquímicos en la facultad, formó muchas generaciones por muchos años, así que chiquillos, quiero felicitarlos, que todos nos pongamos muy felices por esta oportunidad y que todos colaboremos y ayudemos en esta muy linda iniciativa que están formando los bioquímicos de la Universidad de Santiago. Así que muchas gracias.
Le damos las gracias a usted, profesor Felipe. Los miembros de este comité consideran necesario y pertinente rendirle un homenaje a un profesor que significó mucho para la facultad y para sus estudiantes. Le reiteramos los agradecimientos. Les queremos comentar sobre nuestra campaña solidaria mencionada anteriormente de beneficencia, la cual estará apoyada por 3 agrupaciones de ayuda humanitaria en la región de Santiago: el Bosque Ayuda, Peñalolén Solidaria y la Olla de Puente. Nuestra campaña estará recibiendo donaciones voluntarias destinadas en ayuda de las familias de dichos sectores. Les compartimos los datos de la campaña y animamos a quienes puedan contribuir en donaciones o divulgando la campaña a través de sus redes sociales para generar la máxima ayuda posible. Pues bien, para dar inicio a nuestra charla inaugural, queremos invitar a la doctora Alina Baum, bachiller en Ciencias, mención genética de la Universidad de Minnesota, doctora en Biología Molecular de la Escuela Icahn de Medicina de Mount Sinai, Estados Unidos, con estudios postdoctorales en la Universidad Rockefeller de Nueva York. La doctora Baum es actualmente científica senior en Regeneron Pharmaceuticals, sitio donde se ha desempeñado desde 2015, compañía líder en biotecnología a nivel mundial, y que inicialmente se enfocó en el estudio de factores neurotróficos y sus capacidades regenerativas, de donde proviene su nombre, pero con el tiempo ampliaron sus investigaciones al desarrollo de medicamentos para tratar trastornos y enfermedades como el cáncer colorectal, la artritis reumatoide, el ébola y actualmente el COVID-19. Agradeciendo su participación en este congreso, nos presentará su charla titulada Drug Development at Warp Speed, Development of Therapeutic Antibodies for SARS-CoV-2 Virus. Le damos una cordial bienvenida. Ms. Baum, are you there?
Can you hear me now?
Yeah, we can hear you.
Okay, good. Can you see me?
No, not now. We are checking.
Oh,
yeah,
okay.
we can see you right now.
Okay, all right, that's good. This is always the trickiest part that I worry about.
Yeah, we get it.
Computer issues. So thank you so much for inviting me to give this talk. I'm gonna try and make sure that I can present my slides. So let's see.
We are gonna give a little phrase to say that this is going to be in English, and for the questions at the end. Les queremos recordar que la presentación de la doctora Alina Baum será en inglés. Quienes deseen realizar preguntas a la expositora al final de la jornada pueden dejarlas anotadas en el chat de Zoom que aparecerá al final de la charla. Para los miembros de nuestra transmisión en vivo en YouTube, también pueden pueden realizar preguntas en el chat de YouTube, donde miembros del comité estarán compilándolas. Miss Baum, you can start whenever you want.
Okay, can you tell me if you can see my slides?
I can see it.
Okay. All right, great. Well, again, thank you so much for the invitation. What I want to talk about today is I want to tell you a story of how we at Regeneron have been, going at a speed that's, I think, as fast as humanly possible to try and develop antibodies for both treatment and prevention of COVID-19 infection. And before I start talking, so I'm going to be clicking through, before I start talking about our COVID-19 program, I wanted to give an overview of Regeneron. I'm assuming most of you in the audience are not that familiar with the company, and if you've heard of it at all, and give you an understanding of how we are working on these different programs to address emerging infectious diseases, and that we have a history of working on other programs outside of COVID-19. So I want to give you an overview of our overall effort to address some of these big infectious disease concerns. Now, one thing that I really want to start with is to explain, so Regeneron is really, it's an antibody company. We make antibodies for all sorts of different diseases, for different indications, from eye disease to cancer to cardiovascular disease. But we also have an infectious disease program, and Regeneron, it's about a 30-year company, and we've had this infectious disease department for probably close to 15 years. And while a lot of other companies have been leaving the infectious disease field, Regeneron has stayed committed because we realize that this is a really big global concern, and this is something that we're all witnessing right now. Now, one thing about antibodies is that there are certain advantages to antibody-based programs, and this is what is described on the slide here. One thing that's important is that for antibodies that target infectious agents, there's often really good safety profiles. So these antibodies do not bind to anything else in our bodies, and therefore we in general have very little safety concerns about developing antibodies for viruses as well as other pathogens. Now, the other thing that, and one common question that people ask is for a prophylactic setting, so where you're trying to prevent disease, what is the difference between an antibody prophylaxis versus a vaccine? And the answer to that is that they both do, in theory, they do the same thing. They try to prevent the virus from getting in your body and replicating. But the difference is that a vaccine requires a person's own immune response to get activated in order to produce these antibodies, and then these antibodies are protecting against infection. Versus if you make this antibody as a drug, you dose a person with the antibody, that antibody is active immediately. You do not need to wait for a vaccine response to generate the immune response that will be protective. And the other thing that's important is that for some individuals that do not respond well to vaccines, either because of safety issues or because they may have immune systems that are just not that great at responding, for example, the elderly, vaccines may not offer full protection, and that's another place where antibodies could potentially be useful. But in general, we're thinking of these two entities, vaccines and antibodies, as being able to work together, and there may be different places where you can use one versus another. One important thing is that with a vaccine, you try to get a long-term immune response. And in a perfect setting, you could vaccinate somebody once and have that vaccine last for a lifetime. Those types of vaccines are very rare. Usually people need boosts. We have no idea yet how the vaccines that are being developed now for COVID, how long they will work, how well they will work, how often people need to be dosed. For antibodies, you're protected as long as you have enough drug in your system. The drug does get cleared out eventually. So you're not going to be able to dose somebody once a year, for example, with the antibody. But with a really good potent antibody, you could potentially protect someone for a few months. And again, we do not know yet what this is going to look like with COVID because the data have not come out yet from clinical trials. Okay, so on the next slide, what I'm showing you here is just a summary of technologies that we have developed at Regeneron that all work together to develop these fast responses for emerging infectious diseases. What we have are engineered mice that we can put in whatever human gene we want to. And that becomes really useful, for example, when some viruses that infect humans do not infect mice, because the mice could be missing a key factor like the receptor. And this is actually exactly what happened with SARS-CoV-2. SARS-CoV-2 does not infect normal mice. So what we have done at Regeneron is we have made what we call a humanized mouse, which is a mouse that expresses the ACE2, which is a receptor for the virus, from humans. And we can use these mice now to do in vivo assessment of our antibodies and see how well they can protect work in vivo. So that's one technology that we have. The other technology that we have is what we call velocimmune mice. These are the backbone of how we make antibodies. Velocimmune mice are mice that contain DNA to make fully human antibodies. So you may have heard of what is commonly done to get antibodies, for example, for Ebola, for SARS-CoV-2, is people go and they will find individuals that have survived and recovered from the infection, and they will take B cells from those individuals and they will isolate antibodies from those B cells. Now, what we do, we can also do that. We can get antibodies from people, and we did that for this program for COVID. But we also can get antibodies without needing to identify those people, which sometimes can be really difficult if it's a really severe and rare infection, for example. And what we do is we just take these mice and we basically vaccinate them with a protein or DNA that expresses the protein that we want to make antibodies to. And by vaccinating these mice, we can then get fully human antibodies from the mice, same way as you would get fully human antibodies from people. And by doing that, we can get started immediately. So we do not have to wait to get samples from people. As soon as we have a sequence of the pathogen, which is what happened for this program, in January we got the sequence, and right away we made DNA and we immunized mice. And that allows us to move as quickly as possible without waiting for human samples. From these mice, we isolate B cells that are specific for the protein that we're interested in, that are expressing antibodies. And then we go through this process that we call screening, which is basically testing thousands and thousands of these antibodies for properties that we're really interested in. And the other thing that Regeneron does, which allows us to go really, really fast, is as soon as we identify sequences of antibodies that we think are ideal for what we want to put into the clinic, these sequences go into cell lines. Now, cell lines is really, that's how you make an antibody drug. The thing that you make is actually a cell line that encodes genes for this particular antibody. And then you grow these cell lines in these really, really huge containers where you're making, thousands, maybe millions of liters of supernatant that gets secreted from the cells that now contains your antibodies. So we have developed a method to go very fast from our identification of the antibodies that we're interested in to cloning of these cells. And that's really a critical step that kicks off manufacturing, because as soon as you have these cell lines, they can go to the manufacturing facility to start making drug that you can actually then put into the clinic. Okay, so these are the kind of the backbone of the Regeneron technology that that we utilize in order to make antibodies for all of our programs, but also for the rapid response programs. And this is just a cartoon showing an example or more details of how we make these fully human antibodies in mice. Basically, these mice are engineered such as that an antibody has a portion of it that binds to the antigen, which is the variable chain that's circled in blue. That is fully human. But the other part of the antibody, which is the part that's called the FC, and that sits on the cell and signals to various factors that are intracellular, that we retain as a mouse sequence. And the reason that that's important is because now the signaling that occurs between the portion of the antibody that actually has to signal to B cells, and it induces the B cells to mature and to develop and respond to antigen, that is still functional and it can interact with all the mouse components downstream. But the sequence that binds to the antigen is fully human, which is really important. So once this allows us to select for the very best antibodies from these mice, and once we select the very best antibodies, we basically then take that sequence that's human and we put it into a completely fully human backbone. So in the end, the sequence that you get is 100% human sequence. And this is just a cartoon showing basically the same thing, where what you get from the mice is a chimeric antibody. So it has human variable domains, and then it has the mouse constant domain. So it's the blue and the red. But once you go into the making cell lines, you clone everything into a fully human backbone. So now there are no mouse pieces left in it, and it's the same type of antibody that you would get from people. And this is very important for safety reasons, because you don't want these antibodies to be immunogenic, and you don't want them to drive an immune response to the antibody itself, which can happen if your antibodies come from a different species. Okay. The other thing that we do at Regeneron, which also allows us to go very fast, is we, in addition to having research facilities where we isolate these antibodies and identify the best ones, we also have manufacturing facilities. So everything from the very first step in this process, which is really getting the sequence and immunizing the mice, or getting B cells from people, all the way to manufacturing drug that actually goes into our clinical trial and goes into patients. That's all done at Regeneron. So we oversee the process from the very first step to the very last step, which gives us a lot of flexibility. We don't have to find partners to manufacture our drug because we do everything ourselves. Okay. Now, I mentioned that in addition to working on COVID-19, which obviously, the whole scientific community now is working on COVID-19, and every company that may not necessarily have infectious disease programs are also trying to utilize their platforms for COVID-19. The reason why I think we were able to move so quickly and do it so well is because we've done this before. So we have worked on other emerging programs, the latest one being Ebola as well as MERS. And that experience has taught us how to do this in the best possible way. So every time we go through one of these rapid response programs, we learn and we get better and we shrink the timelines without giving up. If anything, the quality of the work that's been coming out has been better and better as well, because, as soon as we start working on these programs, we know exactly who has to do what, and all of these efforts start occurring in parallel. They involve a lot of different groups and a lot of different people, and we just move really, really fast and get an insane amount of data in a short amount of time to make sure that we get the best possible drug into the clinic as quickly as possible. That's really the goal here. For the Ebola program, some of you may have heard, we actually ran the Ebola outbreak that happened in 2018, 2019, and actually there's still cases happening. We were able to run a clinical trial together with the WHO where we looked at the efficacy of anti-Ebola antibodies in treating of Ebola infection. And that clinical trial was very successful, especially for people that showed up to the clinic relatively early in their disease. The antibody treatment greatly reduced mortality that we typically see in Ebola. We're now in the process of trying to get approval for the drug from the FDA. For our MERS program, we also ran what's called a phase 1 trial. This is just in healthy volunteers, so it's not in infected people. But we also developed a combination of antibodies for MERS. And now, of course, we're working on SARS-CoV-2. Okay, I'll give you a quick summary of what we did for Ebola, because it's really a blueprint of how we're also thinking about the COVID-19 program. So this is just a table that shows in a simple way, properties of Ebola antibodies that went into the clinical trial. For Ebola, similarly as SARS-CoV-2, we're developing a cocktail of antibodies, meaning that we want 2 or 3 different antibodies that are going to be given at the same time. The reason for that is we think it's very, very important for viruses that mutate quickly to have antibodies that bind to different patches on the viral protein, so that if a virus develops a mutation, either just by random mutations that come up over time as the virus circulates in people, or potentially even a mutation that's driven by your drug, that by having more than one antibody binding to the protein, you now have the virus has a much harder time acquiring mutations that will make your antibodies no longer effective. So we believe, and different companies are doing different things. Some companies have a single antibody in the clinic. We have a combination of antibodies, and both for Ebola and for SARS-CoV-2. We think the combination is really critical. And so for Ebola, the drug that we had in clinical trials is a combination of 3 different antibodies. And all 3 antibodies have different properties, and they bind to different parts of the viral glycoprotein. So that's the protein that's on the surface of the virus that it needs to get into cells. And this table just summarizes their function. So they neutralize the virus, meaning that they block viral entry into cells. And they also do what we call effector function, which is an antibody that binds to the viral protein that's expressed on a cell that's already infected. And that allows that antibody to bring in immune cells through its Fc receptor, and the immune cells can now kill the infected cells. So that's another property, antiviral property of antibodies that can also be really important for efficacy. So our Ebola antibodies do all of these things, and each antibody does something slightly different. And this just shows you the structure of how they bind, again, highlighting the fact that 3 antibodies, so the purple, the green, the orange, these are 3 different antibodies. And when you look at them all together on how they're pictured on the viral glycoprotein, you can see that they're all binding to different spots. So again, this was really important. These antibodies then, once we had them and we picked these as our leads, that's how we, when we screen thousands of them, eventually we come up with what we think are the very best ones. And the next step is really to put these antibodies and see if they can work in an animal model of the disease that you're trying to treat. So this is what you can see on the next slide. For the Ebola program, the field really uses this rhesus macaque model. These animals are very susceptible to Ebola disease. This, of course, has to be done in a BSL-4 facility. The animals are infected on day 0, and then we treat these animals with different doses of our combination cocktail. And in the red, you can see the animals that don't get treated with anything. They die fairly quickly from the infection. It's a very severe infection that comes in people. And then the other colored lines you can see are the survival of the animals that are treated with higher and higher doses of the drug. So with the higher doses of 100 mg per kg or 150 mg per kg, which are the blue and the black lines, you're now starting to see that you went from 0 animals surviving to 8 out of 9 animals surviving. So we saw a dramatic impact on survival in this animal model. But with like any animal model, you ask, is this really relevant to the human disease? There are always caveats, and no animal model is a perfect model. They're always going to be differences between human disease and whatever you're trying to mimic in vivo. So the only way to really answer that question is by doing a clinical trial, and ideally a controlled clinical trial. What happened last year, which was pretty remarkable for something like Ebola, is that the WHO and the NIH were actually able to run a clinical trial in the DRC during an outbreak in high, areas with political conflict and violence. So it was pretty remarkable effort, but they were able to run this trial and to test 4 different types of therapeutics had to show how different their impact was on survival. So for Ebola infection, typically the survival is somewhere around 50% for people that are just treated with standard of care. What we were able to show, and this is the data from the trial. You can see it. So in the red circle, that's Regeneron EB3. That's our drug. In this trial, the efficacy was measured. There was no placebo group because it's not ethical to for Ebola. It was considered not ethical to give people nothing. It was thought that some of the other drugs that were already tried in the previous outbreak had some efficacy. So in this particular trial, the efficacy was compared to ZMapp, which is another antibody drug. And you could see that the patients are broken up in 2 groups. Here, there are patients with high viral load and patients with low viral load. So patients with low viral load, the survival rate with our drug was about 90%. That's really really remarkable for a disease where a normal survival rate is about 50%. Now, for people that already had more severe disease and already had more virus and were infected for longer, the results are not as great. So for those people, the survival rate was about, it's closer to 50%, but without treatment, those people really don't survive at all. So even though it wasn't as good as in the people with a low viral load, it's a huge clinical benefit even for that group. So at this point, the trial has been completed. It was actually stopped early because of the dramatic efficacy that they were seeing. And we're now in the process of trying to get approval for this drug. Okay. So now I've given you a little bit of a story about Ebola. I'm going to switch to SARS-CoV-2 because that's, and it was actually funny because for Ebola, the trial ended last fall, and in December, we were kind of done with the trial. We were filing our application for approval, and it was a really intense program, we were getting to the point where we felt like, okay, now we're done with this. Now, we're going to take a break and work on other things. So we work on a lot of other stuff that's not emerging infectious disease. And it was like, okay, we can get back to these other programs and start focusing on things maybe we haven't been able to focus on so much because of Ebola. And then, of course, in January, late December, we started hearing, like the rest of the world, of these news stories coming out of China with a few dozen people that were sick with a virus that was similar to SARS. And these things happen. You think sometimes you get these little outbreaks and then the virus kind of disappears or it gets controlled. So at the time, I don't think anyone really could foresee what it was going to turn into even like a month later. But because we do have this rapid response program at Regeneron, as soon as we started seeing news reports that there were dozens of cases and the case numbers seemed to be going up, we were like, okay, it makes sense for us to go ahead and start developing antibodies for this program because that's kind of the whole point of rapid response. You have to go really quickly, so you can't sit there and wait to see how bad the disease is going to get, because that, you know, that takes time. And if you want to roll really quickly, you have to start very early. So we started working on the COVID-19 program in January. Again, as soon as we got the sequence of the spike protein, which is really the key thing that we need to start immunization of our mice. And as we were working, you know, January, it didn't seem like that big of a deal. February, it became quite a bit more concerning because people started picking up cases in other parts of the world. So cases started popping up in Europe. In New York, where we are, it was actually, we didn't have a case for quite a while, which was surprising because New York is obviously a big global hub. And you expect that there was so much travel, there would be cases here, but they weren't picking up cases here. But we were still moving very quickly. And then, you know, some point between like early February and end of February, I think everyone realized just, or not, maybe not everyone, but like a lot of us realized that this was not likely going to go away. That now the virus was spreading in many parts of the world, and it was going to become very, very difficult to get rid of this virus. So at that point, you know, as we were moving through February and March, a virus that at first seemed to be really far away, you know, from us, and it was an outbreak in China, and obviously we want to help, but it was far away. And all of a sudden, it came home, you know, to us. So in March, as we were moving from early March to late March, the life in New York completely, you know, it just transformed. It was like 180 degrees because we went from, you know, no cases to a couple of cases to all of a sudden we knew that we had community spread and it was getting really bad. And in mid-March, the New York basically shut down. Now at Regeneron, because we are, you know, people are working in the labs, people have to come in. So even though the world around us was really shutting down, we couldn't do that. We had to keep going. People had to keep coming into work. And it was a scary time at the time because the people started dying. It was going up. You know, numbers were going up dramatically at the time. Nobody was wearing masks. It was really chaotic. But nevertheless, you know, people understood that we don't really have a choice. So we were trying to be as safe as possible, but we were still working. People were working very, very hard to get these antibodies identified and move them into the clinic as quickly as possible. By middle of April, what we were able to do is to identify the antibodies that we thought were the very best ones. And at that time, as soon as we made that decision, we started manufacturing drug for clinical trials. Because we were able to move so quickly, we were able to initiate our clinical trials on June 11th. So we have a number of clinical trials that are currently ongoing, and I'll mention them again at the end. But these are basically our timelines. It took us, it actually has more dates on this slide. So you can see that from the time we immunized the mice until we had antibodies in manufacturing, it took us about 70 days. We also isolated antibodies from human patients that were infected and then recovered from COVID-19. And for those antibodies, it only took us 33 days. So at the end, we selected one antibody from mice and one antibody from humans. And these two antibodies were put together into a cocktail that we call Regeneron-CoV-2, which is currently being tested in the clinic. Now, the goal for us for the, you know, when you say like, you know, identify antibodies, the key thing you have to figure out is what type of antibodies do you want? What properties do you want them to have? As I said, we screen thousands and thousands of antibodies, so you have to really know what you're looking for. And the key thing for us was to identify antibodies that were really, really potent at neutralizing the virus, which just means that if you put them these antibodies plus virus plus cells into a dish in vitro, you can show that at really, really low concentrations, your antibodies can block the entry of the virus into the cells. So that's what we call neutralization, and that's a key property that we're looking for these antibodies. And the other thing, again, as I mentioned, that was really important is for us to find a combination, which means that we're looking for two antibodies that bind to the spike protein of SARS-CoV-2 at the same time, and they do not block each other. And I'll show you what that looks like in a couple of slides. Now, this is just a background slide with a lot of text, but I find it really interesting because I went, as I was putting these slides together, I went and looked at the talk that I gave in April. And this way you get to reflect on how much have things changed in the last 2 months? How much have we learned of what we didn't know back then? And as I was looking at this, and I was reading, like, what we know, what we do not know, that hasn't changed very much. What do we know? We know now very well. There's no question that SARS-CoV-2 is the virus that causes COVID-19 disease. We know that this virus most likely came from bats sometime in late fall in the Wuhan region of China. We now know that by January, maybe even earlier, this virus was already spreading globally. We didn't know it back then, obviously, because we weren't looking for it. And we know now that because a lot of individuals are either asymptomatic or have very mild symptoms, that this virus is really positioned, almost perfectly to drive spread without being caught. It's very different, for example, for something like Ebola, where the disease is so severe that you don't worry so much about people walking around infecting each other without knowing that they're even sick. Here, many people are completely unaware that they're actually infectious. And this was obviously a huge problem in early 2020. And that's why the cases started spreading like wildfire globally. Now, one thing is the numbers always change. So as I was looking at my slides from April, I think I had 3 million here, saying we have 3 million cases. Now we have over 18 million confirmed cases globally. We know that the real numbers are magnitudes higher because obviously not everybody who's infected is getting tested. And as high as these numbers sound, I think it's also really important to understand that we are still, in terms of the global populations or population in any country, we are very far away from herd immunity. So the vast majority of human beings are naive to the virus. And we're nowhere near like 60 or 70% infection rates that would really take us to something that we can call herd immunity, where the virus slows down and stops spreading so much. And I think that alone kind of makes it obvious that the only way that we're going to be able to go to any kind of normal life is with a successful vaccination program. Now, what that looks like at this point, nobody knows. We don't know how the vaccines will perform, although I think we will get data we start getting data on efficacy hopefully this fall from different clinical trials that are going on around the world. And now we're going to start learning bit by bit, and we'll see. Maybe if everything goes extremely well, maybe next year looks significantly better than this one. But I think at this point, there's still really no way of knowing what the future holds, which is a very, I think, a strange thing for all of us to deal with. This virus, again, it's very similar to SARS, the original SARS virus, so SARS-1, in terms of sequence. They're very close to each other. We have like 80% sequence identity, but the nature of the disease is significantly different. And because with this virus, you do have so many asymptomatic cases, and the symptoms really show up after the virus has peaked in its titers, you do get a lot more spread with this virus than we ever saw with the SARS-1 virus. The scientific community has gone incredibly fast. It's been absolutely amazing how much literature has come out already. It's impossible to keep up at this point with all the findings that are coming out in this field. But we still don't know the answers to some really, really critical questions. We still don't really understand what is the actual mortality rate of this virus, because that really depends on, are you capturing all the infections? Initially, mortality rates were reported to be 5 or even 10% in some places. Most likely that's much, much higher than it really is, because we didn't know that so many people were infected that really didn't have symptoms. But we still don't know the answer to this question. Will it be something like influenza, or is it going to be something that's very different? We don't know if immunity is going to be short or long-lived. It's obviously a very important question, both for immunity to the actual infection as well as the vaccines that are going to be coming out. We don't understand the biological reason of why people have such a different breadth of symptoms. Why are some people barely affected by infection, and then some people have extremely severe disease that results in them either having to be ventilated or death. Doesn't seem like it's the amount of virus that they have. Asymptomatic people have just as much virus replication as people with severe disease. So it likely has something to do with how the immune system responds, and we don't understand the mechanisms behind that. We don't really understand why the disease is so mild in children. It's very unusual for this type of virus. Usually, young children are more susceptible than other age groups. We don't really understand why mortality is higher in men versus women, and so on and so on. So despite all the effort that's being put into research, we're still very far away from having a real understanding of the complexity of the disease that's being caused by the virus. And again, as I was looking at my older slides, I pulled this slide from a presentation we gave on February 12th at Regeneron. And just looking at how the world has changed in 6 months I think it's a really, it's just kind of, mind-blowing to think about what impact this virus has had on the globe. There's probably not really almost no person that hasn't been affected by it one way or another, which is kind of astounding to think of this tiny little organism being able to affect us in so many different ways. And we really, who knows what this map is going to look like in October, for example, or December, or next summer. We don't know what the future holds. So in terms of the biology, again, we know that the receptor for the virus is ACE2. We know that this receptor is expressed in human airways and upper airways and lower airways. And this infection is mostly a respiratory tract infection, but of course, there are many people who have manifestations that are external to the respiratory tract. And we know now that problems with the circulatory system are common in severely infected patients, and people have reported all sorts of issues with their kidneys, neurological issues, skin disorders, and we really don't understand the reason for that and why that happens in certain individuals. This is just a slide showing the cartoon of the virus and the viral life cycle. Now, I'm sure everyone has seen this picture of the virion covered in spike proteins. The spike protein is really the key thing that a lot of the therapeutics and vaccines are targeting, because that's the protein that the virus absolutely needs to get into cells. And if you can block that interaction between the spike protein and the receptor that it binds to on the cell surface, you can block the virus from getting inside the cells, replicating, making more copies of itself, spreading, and making somebody sick. So essentially, every single vaccine that's being developed is using the spike protein one way or another as the antigen. And the antibody programs are also targeting the spike protein because that, again, is the most efficient way to stop this virus from infecting cells. This just shows a diagram of how, on a structural basis, how the spike protein actually interacts with the ACE2 receptor. It's an interesting structure because it has, so in the red is this whole, the whole picture, that's the spike protein, but in red is what's called the receptor binding domain. And that's the part of it that interacts with ACE2. And this receptor binding domain, it kind of opens and closes, and it goes in this open conformation. This open conformation is what it actually needs to bind to ACE2. When it's closed, it cannot bind to ACE2. So that interaction, and the open one is shown in the green in these cartoons. So that interaction between the open receptor binding domain, or RBD, and ACE2 is what we're trying to block with our antibodies. Again, because that is the process in the virus life cycle that's absolutely required for it to get into a cell and replicate. Which it does, as you're looking from A all the way to F, that's the progression of how the protein binds to ACE2. And when you see in D, for example, that's now it has all the RBDs are open and they're binding to ACE2. And when that happens, what you see is this dramatic change in shape. So it goes from what looks like the stalk with the RBD on top to that shape that's in black that now looks like a completely different protein. But it's still the same, it's the same spike protein. It has now just gone through a conformational change. And that change is what's needed for this protein to fuse the viral membrane to the cellular membrane, which is really how these viruses work. They fuse the two membranes together, and then the virus can deliver its genome to the cell cytoplasm, which is really where it needs to go in order to make more protein, to replicate, to make more virus, and eventually bud out of that cell and infect new cells. So now, I mentioned previously that what we were looking for is what kind of antibodies we want to make. We want to make antibodies that neutralize the virus, meaning they block virus entry. Now, one thing that we've had to deal with, and what a lot of researchers on SARS-CoV-2 have to deal with, is that this is a BSL-3 pathogen, which means it can only be worked in laboratories that have a certain biosafety level. And we're not actually using this virus at Regeneron. What we did, though, is we used virus engineering to make what we call pseudoviruses. So this is basically taking one virus, which here it's called VSV. So VSV is a virus that you can work with in any BSL-2 level lab, which are most labs that we have. And you can take the native glycoprotein from VSV, you delete it from the genome, and you can replace that with a spike protein of SARS-CoV-2. And you can do this in a couple of different ways. You can make replicating virus, that's a chimeric virus, or you can make a non-replicating virus, which is basically now a virus particle that contains a spike protein on the surface. It can enter cells. It can deliver what we call a reporter, which is something that gives you a signal. So for example, it can be GFP, so the cells turn green, or it can be luciferase, so we can measure luminescence. And that way we have a system to look at infectivity that's mediated by the spike protein through the ACE2 receptor, exactly the same way as it happens in a natural SARS-CoV-2 infection. But now we have a system that can be used in a BSL-2 setting versus a BSL-3 setting, which makes a huge difference because we're not restricted to these biosafety labs that are not particularly, extremely common. So by using this VSV system, that's exactly what we did. And we now have a system where we can measure the infectivity of these particles, and more importantly, we can measure the potency of our antibodies at blocking the infectivity, which is exactly what you see on this slide. So on the left-hand side is a picture of what I just mentioned. We take these VSV particles that contain a green reporter, and when you infect cells with this is what you see. You see these green dots everywhere that tells you the cells are infected. If you take an antibody, what you do is you just dilute that antibody and you add the dilution of antibody to your wells at the same time as you're adding the virus. And now by looking at whether or not you have the green color, you can tell whether the cells are infected or if your antibody was able to block the infection. So that's what you see here at the higher concentration of antibody on the left-hand side. You no longer see the green. So that tells you that the antibody has blocked the infection. And by measuring the concentration at which these antibodies are blocking the infection, we can calculate the potency of the antibodies. So for our program, what we were able to do in order to pick our leads that go into the clinical trial is we actually did this with thousands of antibodies. So this is run as a high throughput screening assay. And we test thousands and thousands of antibodies, and then we pick the very best ones. And the best ones, or some of them, you can see on the right-hand side of the slide. So these are our neutralization inhibition curves, where you can basically look at, the exact concentration at which your antibody is neutralizing 50% of the virus. That's the IC50, and that's how everybody compares potency of these antibodies. So the y-axis is percent neutralization, and you can see that on the right-hand side, you're at 100% neutralization. As you dilute your antibody out, eventually you get to 0% neutralization, and then you just measure the concentration at which you neutralize half of your virus. That's your IC50. So this has probably about 10 of our best antibodies that we identified in our screens, and this is the list where our clinical antibodies came from. The other thing that we did while we were selecting our leads, thing that was really important to us, and I mentioned this before, SARS-CoV-2 is an RNA virus, so these viruses are known for mutating quickly. They will acquire random mutations just through replication. So we wanted to make sure that the antibodies that we put into the clinic are going to work against the viruses that are circulating globally. And in order to do that, what we were doing the whole time as we were developing these antibodies, we were also looking at sequences of all the patient isolates that we're getting coming in through around the world. We were looking at mutations in the receptor binding domain to which our antibodies bind. And by using the same virus engineering system, we were able to make spike protein that contains these mutations. So we could test whether any particular mutation that you identify in people will actually make our antibodies no longer work. And so obviously, again, that's really important because you don't want to spend months making a drug and then find out as you're going into clinical trials that now the virus has mutated and your drug no longer works. That would be, a really bad scenario. So what you see in this table is just a list of mutations. Now, this list came from April. We're continuing to do this. We're going to do this for as long as we have these antibodies anywhere in the clinic. So we're making more and more mutants because more and more mutants are being identified in people, and making sure that the antibodies that are in the clinic still have the same potency that they have for what we call wild type virus. And that's just what table shows. It shows that we have potency against these different mutations. We also, because we did want to identify antibodies that bind to different regions of the RBD, as we were getting closer to lead selection, we picked some of our best neutralizers, and we were trying to identify patches on the spike protein where these antibodies bind. We were doing this before we had high resolution structure of these antibodies. So this was a quick way for us to get an idea of where these antibodies bind, because if you want to get a combination of two antibodies that don't bind in the same place, this gives you an idea if your antibodies bind to one region or another. And you can tell from, from these cartoons, these different colors just give you different regions where some of their antibodies were identified to bind. And some of them bind in one place, some of them bind in another place. And this gives us like some structural information before we actually get at structure. The other way, which is really important, again, for identifying a combination is by running what we call cross-competition assays. This is just an assay where you take one antibody, one of your antibodies, you bind it to the receptor binding domain, and then you see if you can now bind the second antibody to the receptor binding domain where the first antibody already bound. If these antibodies compete, the first antibody will block the second antibody from binding. If they don't compete, they can both bind at the same time. So that, again, is a critical component for selection of the cocktail. And the table here just shows the outcome of this experiment, where basically you can see a list of antibodies that are on the left-hand side. It's the same list that's going across. And you can see how these antibodies pair up. So the colors that are red tells you that these two antibodies are blocking each other. And the colors that are white tells you that these two antibodies are not blocking each other. So as we're going through the selection process, we're eliminating the pairs that give us the red color, and we're focusing on the pairs that give us the white color. And you can tell that actually most of these antibodies compete. So it's not trivial to identify two antibodies that don't compete and that also have really high neutralization potency. We really need to screen thousands of them to get to that combination. But that's exactly what we got. Here you can see we were able to actually get the cryo-EM structure of our antibodies. So the two antibodies that are in clinical development are called Regeneron 10933 and Regeneron 10987. And here you see the structure of how they bind to the spike RBD. And when you look at this, it becomes completely obvious why these two antibodies don't compete with each other, because they bind to completely different portions of the protein and they bind in different orientations. Now, the last thing that I'll talk about, which again was something that was very important for us to do, is in addition to looking at natural variants of the spike protein and sequencing those and then looking to make sure antibodies work against that, you can also drive selection of what we call escape mutants. So these are mutations that don't arise just because of natural replication of the virus. They arise because you're putting pressure of your antibody onto the virus and you're forcing this virus to mutate. The way we do this is we still use the same VSV system. So it's VSV virus that expresses the spike protein. This virus, we infect cells in the presence of antibodies. And what happens as this virus is replicating in the presence of antibodies, these viruses exist as a population that has a bunch of different mutations in it already. If you have a mutation in there in the spike protein that makes that particular virus not be neutralized by your antibody, that particular, so that's the orange guy in this cartoon. So while all the other variants of your virus are neutralized by the antibody, the orange guy isn't. So the orange guy is the only one that can grow in the presence of antibody. And eventually, this orange guy will take over the whole population because all the other versions of the virus are neutralized. So this is what we call selection of escape variants. And the reason we want to do this is because we want to test our antibody combination and see how easy or how difficult is it to get escape. What you worry about is this situation happening in a person. So instead of doing this in a tissue culture dish artificially, you can have a similar thing happen in a person where the virus is replicating, you have your antibodies there, and your antibodies are putting selection pressure for your virus to evolve and select for the versions that are resistant to your drug. And of course, you do not want that to happen because then this resistant virus can spread to new people. And if that happens efficiently enough, all of a sudden you have a drug that no longer works. And we know that this is something that's a serious concern in real life, because this has happened with many drugs that were developed for various infectious diseases, and HIV is a very good example, where some first-generation antivirals, very soon the virus became completely resistant to all of them. This is obviously, an issue with antibiotics as well. So this is really the last slide with data, and this just shows what what the readout of these experiments look like. One thing we were doing in these experiments is we were comparing a single antibody versus a combination of two non-competing antibodies. And the way to interpret this data, the way this is done is we take, again, this VSV virus, we add this virus to cells with a dilution of our antibodies. So when on the right-hand side, you can see at the top, it's showing you antibody concentrations. We go from really high concentration all the way to no antibody on the right-hand side. And the blue here shows you that the antibody is working. So it's blocking the virus from infecting cells. The red shows you that the virus is now infecting cells, so these cells are dying. That's how you read out the outcome of this assay. And the important thing is just to look at the shift from the red, from the blue to the red, because a shift from blue to red means that you have escape. So for example, as you're going from passage 1, which is at the top, to passage 2, which is on the bottom of this plate, you can see there are 4 individual antibodies, 10989, 87, 33, and 34. Again, 87 and 33 are clinical antibodies, but here they're being used as a single antibody. And for all the single antibodies by passage 2, which is the bottom plate here, you basically had every single one of these antibodies selected for an escape mutation. So this happened very quickly. And the virus was able to evade these antibodies, and now you have a version that's resistant to antibody treatment. Now, if you compare that to what happens when you have a combination of these antibodies, which is at the at the bottom of the plate, you can see 10987 plus 10933. So now you've combined them together into the same wells. And this virus can no longer, escape. So at these, in passage 2, whereas your individual antibodies are completely losing efficacy because of the escape means, the combination still potently neutralizes your virus. So again, we think this is really important because we do not want to have a scenario in the clinic where we have potential escape of the virus that's resistant to the drug that's being used for the disease. So lastly, again, I mentioned in the beginning that we have clinical trials ongoing. We are testing this virus, or we're testing our drugs in 3 different settings, all the way from, prophylaxis, which is, can we prevent disease in someone who we think might be, might either be exposed or might be at high risk of being exposed, to can we treat someone? What we call ambulatory is a clinical trial that's focused on patients that are not severe enough to be hospitalized. So these are people that come in, get tested, they're diagnosed with a positive PCR test, but their symptoms are not severe enough to hospitalize them. So we want to see if we can treat these people and minimize some of the potential, consequences of negative consequences of COVID-19 disease. And we're also running a trial in patients that are hospitalized. So these are people obviously with more severe disease. Most likely they've been infected longer. But obviously that's a huge need that's probably not going to go away for quite a while. And we really want to be able to offer something to people that are have severe disease and require hospitalization to see if there can be a benefit in that population from an antiviral treatment. So these studies are ongoing. We are expecting readouts from these studies, hopefully, sometime either in late summer or maybe early fall. It's hard to know yet exactly when, but this is really the most important thing. All of this work and everything that we've done so far is really just leading up to this moment. And the outcome of these trials is going to be the most important thing that we're going to learn of whether or not antibody therapeutics are really going to be effective in this setting. So we're very anxiously waiting for that data to come out. We don't know the answer to that yet. And just to put things in perspective, because it's always kind of amazing to me what the effort actually looks like to do so much work in such a short period of time. It really takes, hundreds of people to get this program to where it is. So this is just the list of the people in research. So this doesn't even include people that are doing the clinical work and regulatory work. It's been a really remarkable effort by literally hundreds of people to pull this together and pull it together so quickly. The program for COVID-19 is supported by BARDA, which is a US agency that funds pandemic preparedness. So we're working in close partnership with them for the clinical development of this program. Okay, so with that, I will stop projecting. Okay.
We would like to thank you, Ms. Baum, for your presentation. And we have a member of the group who will take the questions that are in the chat.
Okay, great, thank you.
Damos paso a una ronda de preguntas. Invitamos a Felipe Gatica, miembro del comité organizador, quien leerá las preguntas recopiladas.
Yeah. Good afternoon, dear Alina. I have the ultimate pleasure to communicate with you. But first, I would like to thank you for your time, your great talk, and your work. So I'm going to start with Frank Roth, Dr. Frank Roth, which also is one of our part of the Congress. He has the ending talk. So I'm going to read the question. Frank Roth says, I have the question about the reports about MMR vaccination may be protective. Is there any information on the specificity of MMR antibodies versus COVID proteins? Another question, can you devise a map that blocks AC2 and occludes it versus viral binding?
Okay, so those are both interesting questions. I think in terms of MMR being protective, I am pretty skeptical that that's something that, they can really be protective. We saw a lot of reports about the BCG vaccine also potentially being protective. It's possible that there could be some kind of non-specific immune response in recently vaccinated people, for example, that could give you some low-level protection, but the viruses are so different that it's impossible to imagine that there would be antibodies or T cells that would be cross-reactive between the MMR vaccine and the COVID-2 vaccine. And I honestly haven't seen any really convincing data showing that to be the case. So personally, I would be pretty skeptical about that. In terms of an antibody to block ACE2, yes, that's a good question, and it's definitely been considered. There are a couple of reasons why I think anti-spike antibodies are, well, one, we know that they're more effective. So we know that they have much better potency than just blocking ACE2. The other problem with blocking ACE2 is you also have to worry about the function of ACE2. You don't want, ACE2 seems to be important for actual potentially protection against lung damage. And if your antibody is going to be somehow also impacting the ACE2 function, now you have a safety concern that you do not have with anti-spike antibodies. So we think that targeting spike is just more efficient and safer way to go after the virus.
Thanks. Another question. Jorge Bolton, which also is a part of the crew that organized the Congress, asked, what is your point of view about the people who think that the pandemic is a conspiracy? What can we do in relation to this situation? That's an interesting one.
Well, I think the important thing is, you know, obviously people that think that this is a conspiracy theory, the main question to me is how do you deal with that, and how do you address that? And it has to come back to science communication, and just speaking the truth as much as you can. It's very unfortunate, and it's been a different scenario in every country, and it's because this is a global crisis. It's actually kind of interesting to see how different countries are responding, because for something like this, you really require leadership to set the tone. And it's up to each government. You know, this is something that I think individual scientists, we can speak the truth, we can talk, we can communicate, but that's not enough. You really need this to happen in every level of government. And it's, you know, it's very challenging to deliver a message when there are people in power that might be trying to deliver a different message. And I think we're seeing the consequences of that now. So for myself, all I can do is say what I think and look at the data, but we're scientists, and that's what we do. It's hard sometimes to convince people that do not want to believe something for personal reasons. I don't have a great answer to that question. It's a problem.
It's okay. Yeah, we have to deal with Bolsonaro. Yeah, Carolina Leal asks, what kind of expectations can we form from animal experimentation in order for it to work on humans? How many positive outcomes do we need in order to expect it to be successful in humans, considering the difference between species?
Also a very good question. So we're actually about to publish on bioRxiv some of our in vivo data. You know, because this field is moving so quickly, right? There aren't really established animal models. Everyone moved in the spring to test all these different animal models and try to identify which animal model is, I guess, the best surrogate for human disease. And it's complicated because the human disease is so varied. So it's also a question of what type of human disease are you trying to mimic? Are you trying to mimic a mild infection or a severe infection? We have been focusing on running animal models in the rhesus macaque model, in the hamster model, and now, as I mentioned, we also made humanized ACE2 mice that we're testing right now. We are seeing promising results in these animal models, both looking at ability to impact pathology as well as impact on viral loads. But to be perfectly blunt, the only way you can really validate an animal model is when you can eventually tie the animal model data to clinical trial data. So while it's promising and you really want to see an impact in an animal model, and if you didn't see an impact, it would be concerning, because in theory, an antiviral should be able to target the virus in different species, right? We're going after the virus directly. It's a little bit, I think it's more challenging when we're trying to, for example, modulate the immune response, because then it gets more complicated, and different animals are very different from each other. So we are hopeful that animal model data that shows you impact on minimizing lung pathology and minimizing viral loads gives you a clue that maybe that's what you can expect in your clinical trial. But until the clinical trial data comes out, you do not, you just don't know. So that's what we're waiting for, is the clinical trial data.
Yeah, thank you. Yeah, thanks. That was my angry dog. Carlos González Contreras asks, has it been possible to determine a difference between SARS-CoV-2 immunogenicity and MERS or SARS-CoV? How does it relate to possible reinfections?
So SARS-2 and SARS-1 are much more similar than SARS-CoV-2 and MERS. MERS is more different than these two viruses. In terms of antibody responses, you can get cross-reactivity between the two SARS viruses in binding, but most of the cross-reactive antibodies are not neutralizing. So these viruses are differently enough where the portion of the spike protein that binds to ACE2 is actually different enough in these two viruses that your most potent neutralizing antibodies are virus specific, which is why there are some people out there that are testing cross-reactive antibodies they're developing them for clinical use. But what we see when we test the potency of those antibodies is they're less potent than antibodies that are specific for the two viruses. In terms of T-cell responses, there also seems to be overlap with these viruses, and actually, maybe more importantly, because not that many people in the world have been exposed to the original SARS virus. It was a very limited infection of about 8,000 cases total. But there are seasonal coronaviruses that are extremely common and that the majority of the human population have been exposed to. So I think a much more interesting question is whether there's any cross-reactive immunity between seasonal coronaviruses and SARS-CoV-2, especially for T cells, and whether that mediates any level of protection and could maybe also potentially explain differences in severity of disease between different individuals. So we don't know the answer to that yet.
Thanks. Here we go with another one. Nicolás Corrales. Thanks for the very interesting talk. I wanted to ask, how do you do the immunization of the mice once you have the sequence of the protein? As you showed, mice produce neutralizing antibodies. So will this formulation also be a vaccine candidate that could elicit this kind of neutralizing response in the host?
Well, in theory, right? In theory, it could be. We could make the mice are immunized with DNA, then they're boosted with receptor binding domain protein as a protein boost with adjuvant. So these are all typical things of a vaccine. Also, the VSV virus that we use for our neutralizing assays, so this VSV express spike, that's actually also a vaccine platform. So for example, Merck is developing a vaccine that's based on this VSV virus. So in theory, these could all be used as vaccines. And the thing is that Regeneron is not really a vaccine company. So we're focusing on the thing that we do best, which is antibody therapeutics. Other people are focusing on things that they do best, which is vaccine manufacturing, because every drug is, even if you can make it and put it in a mouse and show that it works in a mouse, the process of drug development is extremely complicated. And everything from, getting it approved through regulatory agencies and especially manufacturing, you need to have the know-how to do it right. So it makes sense for companies that have experience in certain platforms to put all of their focus on those platforms, which is why we're focusing on antibody therapeutics, because that's what we know best.
Thanks. Well, we have plenty of questions. So Liesl Friedrich asks, why is it better to use a mixture of competitive and non-competitive antibodies?
So it's much better to use non-competing antibodies because, and actually I didn't really focus on this point because I was trying not to go too long, but we also tested a combination of competing antibodies. And the fact that they're competing means that there's some sites on the spike protein that overlap between the two antibodies. And what we found when we were doing our escape studies is that for one of those combinations, a single mutation actually took out both antibodies. So that kind of defeats the whole purpose of a combination because you want your two antibodies to have absolutely no overlap with each other so that they cannot be taken out by a single mutation. It's much harder for this virus to get two mutations that take out two antibodies than for it to get a single mutation that takes out one antibody. So that's the big difference between those two.
Yeah. Thanks. I think that you brought the question very well, so I understand. Isidora Molina says, how do you think we as a scientist community have to act against the anti-vacciners? Which is the best strategy?
I think getting angry is the worst strategy. Like, so I think that's the most natural response, because as a scientist, you realize how irrational that thought process is. But I also feel I try to be sympathetic, because I'm also, I'm a mother. I have 2 little kids, and I can put myself into this mindset that if you don't really understand something, that it can seem scary. And if you hear, internet is full of like information, misinformation, everything in between. Sometimes when you read an anecdotal story of somebody telling you, what happened to their child, that can resonate much more with people than reading a PubMed article that maybe has all the controlled data and the errors and everything, but that doesn't resonate with people. Most people can't, even if they wanted to and they could understand that, there's no emotional response to that. So I sympathize with, with why. I don't necessarily understand why, and how this whole anti-vaccine movement became so big, but I can understand why a person who maybe doesn't necessarily have a scientific background, it's hard for them to know what the truth is, because there's so much information on the internet, and unless you know better, you can't tell one source from the other, and sources that are completely illegitimate can seem legitimate. Again, I think it's the same question as like, what do you do when someone tells you that the COVID-19 is a hoax? You can try and have a reasonable discussion with someone. You can point out examples. You can, you can tell them what you think. But in the end, I think what we need, it's the same problem. You need really focused government communication efforts to try and communicate to people and explain to people what the reality is. It's really unfortunate that the scientists are not necessarily believed, and I don't know what the perfect answer to it is. I think it's a huge problem. We need more people to go into science communication who can connect the scientists to the public out there, because it's a hard thing to do, and it takes a special skill set to be able to communicate effectively. So I think we need more people that have that skill set to go into those fields, and for those to be supported by governments and different organizations.
Yeah, we need a lot of Carl Sagans over there. Isidora Balgotin asks, says, I wanted to ask your opinion about what the World Health Organization said of the possibility that we will never have a solution for COVID-19, despite the progress some labs have made in the production of a vaccine.
Is that a quote? I don't remember listening. I don't know. I don't remember reading that exact language, so I'll try to interpret, in my own, what I think maybe that means. I think that there's a little bit of misconception by a lot of people that, as soon as we have vaccines that show efficacy in clinical trials, that somehow the problem is over. And that's obviously not how this is going to work. I think people have to realize that even if everything goes amazingly well, that we see efficacy that's real. What does efficacy look like? Does it mean that the virus is completely eliminated from people that are vaccinated? Or does it mean that maybe they're infected, but severe cases drop by 70%? Like, all of those could mean success, right? But if you can minimize the severity of the disease, and it doesn't go away, the virus is still going to be out there. It's still going to be infecting people. It's going to take a really long time, even if vaccines are effective, to vaccinate the world. You know, how are we going to do that? That's a big question. And all of that is going to take a lot of time. So I think that the expectation for people shouldn't be, once we have vaccines that show efficacy, this is over. It's going to be a very slow process, gradual process, hopefully with shifts, you know, in risk. And it's not going to be like one day we can all take our masks off and go watch a football game. I don't think that's going to happen anytime soon. It's going to have to be a combination of vaccination, treatments, and, you know, maintain social distancing, maintain mask wearing, and maybe if we do that right, within a few years, we can go to something that looks real. And maybe this virus never goes away, but maybe it becomes something like seasonal flu, where we just learn to live with it, and we know that there's a certain level of risk, but it's not as dramatic as it is right now. I maybe that's what they were trying to say.
Yeah, yeah. Wow, this is a long journey. Well, Pablo Palacios says, as to the effector function of the antibodies, have you performed a screening employing the different constant regions of the heavy chains of immunoglobulin in the antibodies already developed by Regeneron? In example, alternating between IgG1 and IgG3 in order to study if there is a best suited vector response for a certain pathogen.
Kind of. So the antibodies that we have in the clinic are IgG1. We had to make a decision early on because, you know, as you make a clinical drug, it's not that simple to go back and forth and to change what you're doing. There's a huge effort that goes into manufacturing the drug and putting the drug in clinical trials and going through approval. So we made a decision to use IgG1 antibodies because even though there was very little data on coronaviruses and the role of effector function, there is data for other viruses showing that effector function could be important for efficacy. What we have tested and what we're still trying to understand is comparing antibodies that have IgG1. So IgG1 allows you, the antibody, to bind to a lot of different Fc gamma receptors. This is really what you need to bring in immune cells. What we do is we make antibodies that we call uber stealth. This is our version of an IgG4 where it has an additional mutation that basically eliminates all the binding to the Fc receptors. So we can test these two completely different scenarios. Something that binds to Fc receptors and something doesn't bind to Fc receptors, which allows you to ask the question whether effector function has anything to do with efficacy, and also whether there are any kind of safety concerns associated with effector function. So we are still testing that in animal models, and really that's the only thing you can do is you can test it in animal models. You can see how if there are any differences in animal models, and based on that data, you would then, if something was really concerning potentially, you would decide, well, maybe we need to switch our drug. Now, again, it comes back to like how good are animal models surrogates for human disease. We don't have an answer to that yet for COVID-19, but we are, we have been very cognizant of effector function, and we are trying to, by doing experiments and running these experiments both in cells and in animal models, to understand what role it might play in efficacy.
Thanks. Well, Valentina Hernandez, in your experience and for what you have studied about
No, you're cutting out. You're cutting out a little.
Sort this virus to affect minor.
Felipe, habla de nuevo, te caíste.
Me caí. Ya, perfecto. Sorry.
That's okay. Internet problems.
Yeah. Well, Valentina Hernandez asks, in your experience and for what you have studied about SARS-CoV-2, why do you think children are sort of immune to this virus? Why do they get infected in a minor proportion?
I don't think there's data showing that children are immune to the virus. I think, the data in children is really interesting. And I think part of the reason why we don't know so much is because children are not tested nearly as much as adults. And part of the reason for that is, again, because children don't seem to, thank God, I think we can all say, to be spared from high frequency of severe infections. So there just hasn't been as much focus on studying this virus in children. But with more and more data coming out, it does seem like children do get infected. Potentially, they can even spread as much as adults. It's not clear yet because different studies are showing very different things. And they just don't seem to have the frequency of severe disease that adults do, especially as age starts increasing. And the reason for that is completely not understood at all. I think that's one of the really big questions out there.
Yeah, thanks. Yeah, it's difficult because, as you said, the focus is not in that group of population.
Mm-hmm.
Maximiliano.
Go ahead.
Okay.
I was saying people are now focusing more on that because we're having big debates about schools opening in September. So all of a sudden, there's a lot more focus on trying to understand if children can, at least if they get infected and spread the virus as much as adults, because there's a lot of controversy about what to do with the schools.
Yeah, it's a difficult subject. Well, Maximiliano Urrutia says, "Hi, Ms. Baum. I want to thank you for your presentation and participation in this Congress. My question points to how necessary do you see the option of creating a more established global scientific network to face future global pandemics?" Wow, that's a big one. Despite of the present networks of collaboration and the WHO leadership of Global Health Organization leadership, which is clearly oriented by economic and political interests.
That is a big question. I think in some ways there's already, the scientific community is a global community. So I think when it comes to research, it kind of feels like it's already there because there's so much collaboration across different academic institutions. As far as developing, making drugs, distributing drugs, that becomes an insanely complicated effort. And we do have these organizations like the WHO, obviously, that are there to serve that purpose. But of course, they're not perfect. And of course, it requires participation of governments. In the end, it's the local, the country governments are the ones that have to be part of this, and they have to, they cannot be forced to be part of any kind of a global effort. And for some countries, and this is the case in the United States, even having some kind of a coherent national strategy has been difficult. So it's hard to imagine how you can take that and now make it into a global strategy. I think in a perfect world, that would be the case, but it's definitely outside of my expertise knowing how you do something like that.
Yeah, that's a big one. Well, yeah, it is more clear now. Bastian Rivera says, the vaccine that Regeneron proposed is directed against spike protein. This means that not only allow prepare an immune response before the viral infection would also stop an infection in progress, since it blocks its recognition by AC2.
Felipe, ¿puedes repetir la pregunta? Te caíste nuevamente.
Ya. ¿Me escucho bien ahora? ¿Pablo?
Sí, pero
Ya.
se te ve. Dale.
Yeah, okay. I'm going ahead again. The vaccine, Bastian Rivera says, the vaccine that Regeneron proposed is directed against spike protein. This means that not only allow prepare an immune response before the viral infection, but also stop an infection progress, since it blocks its recognition by AC2.
Maybe, so if I understand correctly, the question is if it's gonna, so first of all, we're making antibodies, right? That's not exactly a vaccine. And if we're using these antibodies to prevent an infection, we are hoping to stop the virus from infecting cells and to stop it from spreading. I don't know if I'm answering the question that you're asking though.
Okay. Yeah, I have another one.
Okay.
Cristóbal Espinosa pregunta. There is a lot of questions here. They're very interested in your talk. That's good. That's good.
I have about 10 more minutes just to.
Okay. Yeah. I think that this will be the last one. So.
Okay.
Yeah. When you perform your antibodies assays in mice, did you take into account its sex? Did you see any significant differences between both genders? And finally, is there evidence about estrogen's role in this?
Yeah, we haven't done those experiments yet. I think they have been done by other groups. And if I remember correctly, I'm not sure that anyone has seen an obvious gender difference in any animal experiments. But again, it could be, it could be something that's a little bit subtle, because if you think about the differences in men and women, the differences are not overwhelming. It's just when you look at, the number of hospitalizations, for example, it's like, well, 55 are male, 45 are female, but that difference has been consistent globally, which is why we have high enough numbers to think that it's really true and there is a gender difference. But I haven't seen data from any animal models that that tries to or shows you some kind of a significant difference. In terms of estrogen, again, I think that's one hypothesis. I guess when you see a gender difference, you could think that maybe that has something to do with it. But I haven't seen any data that actually shows you experimentally that estrogen could be protective. Now, again, because there's so many papers now and so many studies coming out, it's almost impossible for any one person to be aware of every single study. So it's possible that, if you look for this, maybe you will find something, but it hasn't been like something that has come out in the field as potential meaningful explanation.
Yeah. So we finished the part of the questions, and as a part of the team, I gotta thank you again for your time, your disposition. I think that that's the right word. And we will be in touch. I gotta go. Now Pablo is coming, so it's a pleasure.
Okay, well, thank you so much for inviting me. It was really a pleasure to speak, to tell us our story. I know we're very far away geographically, so it was really nice to be able to talk about what we're doing here and to hear the questions and hear from everyone. So thank you.
You're welcome, Miss. Alina, we are very grateful for your presentation and your collaboration with this Congress.
Thank you. Bye-bye, everyone.
Bye-bye. Aprovechamos de recordar a todos los asistentes al Congreso que nuestras plataformas por redes sociales están disponibles para entregar información oportuna del día a día. Allí también podrán encontrar toda la información sobre la campaña solidaria de beneficencia. Los invitamos a seguir nuestras plataformas de Instagram, Facebook y Twitter. Damos por finalizado este primer bloque. Agradecemos enormemente la asistencia de todos y todas y a nuestros socios los estaremos esperando en el próximo bloque más tarde para participar en los talleres que preparamos para ustedes. Esperamos verlos a todos mañana en la jornada de nuestro simposio de cambio climático a las 10:30. Tengan un excelente día, cuídense mucho y nos vemos mañana con más actividades.